Good afternoon, Professor Neelima Gupta, the Vice Chancellor of Tilka Manji University, will be joining us soon.
Check.
Good afternoon, Honorable Madam, Professor Neelima Gupta. Good afternoon, ma'am. Good afternoon, ma'am. Good afternoon, Honorable Ma'am, Professor Neelima Gupta, the Vice Chancellor of Tilka Manji, Bhagalpur University. So, ma'am, please unmute yourself.
Yeah. Is it okay?
Yes.
Fine. Can you hear me?
Yes, ma'am. We can hear you. Yes.
Okay.
This is
Okay.
Dr. Shanti Desai, and we were running the sessions late. So we are in end of one of the session, which was there on the sustainable development in lipid biochemistry. And we are going to have your address now. And we have the chairperson on the platform as Professor Shobna Kapoor from IIT Mumbai. So now I call upon Dr. Avelino Di Costa to introduce you, ma'am, and then you may start your address for this national seminar. Over to you, Dr. Avelino Di Costa.
Thank you, ma'am. It is an honor to introduce Professor Neelima Gupta at this national seminar. Honorable Madam is the Vice Chancellor of Tilka Manji Bhagalpur University in Bhagalpur, Bihar, and she is also holding the additional charge as the Vice Chancellor of Munger University, Bihar. She was also the Vice Chancellor of Chhatrapati Shahuji Maharaj University, Kanpur. She has served as a professor in the Department of Animal Sciences, Mahatma Jyotiba Phule Rohilkhand University, Bareilly, where she has held the positions of head, dean, and coordinator of the Center of Excellence during her tenure. Professor Gupta has contributed to thermomolecular SEM taxonomy and protein profile based on 60 species and 7 gene bank submissions. She is presently working on the pollution status of the river Ganga and protozoan and fish health. She has several international collaborations, such as with University of Shiga Prefecture in Japan, South Valley University, and National Research Center in Egypt. She is a fellow of Zoological Survey of India, Indian Academy of Environmental Sciences, International Society for Ecological Communications, and International Consortium of Contemporary Biologists. She has also been a visiting scientist at the Fish Culture Experimental Station of Poland Institute of Parasitology. She has more than 200 publications and 8 books published to her credit. She is also a recipient of several scientific and academic awards. She is the member of Planning and Monitoring Board at the Mahatma Gandhi Central University, Mothari, the President of India's nominee as member of Executive Council, Central University of Tamil Nadu, and UGC nominee Board of Governors, Gautam Buddha University, Greater Noida, to name a few of her important recognitions. We are privileged to have you, Honorable Madam, at this national seminar on life and life processes. I now humbly request Honorable Professor Neelima Gupta Ma'am to address the gathering.
Thank you very much. It is really an honor for me to be a part of the 3-day international seminar, which is being organized on life and life processes, interdisciplinary approach for sustainable development, which has been organized by the University of Goa. And first of all, I apologize that as per my commitment earlier, I was supposed to deliver this talk on Monday, that is the 19th. But due to my some urgent meeting which had come up, I could not deliver this talk on that day. And you were considerate enough to re-invite me and give me another opportunity as per my availability. So I really feel very honored and gratified for including me in this webinar. I see a number of important people who are associated with this webinar, since you have been holding it in different sessions during the 3 days from 19th July to 21st of July. And I'm sure that all those who have been associated with this webinar, they have been greatly benefited by the talks which have been delivered by eminent people and the active participation of the participants who are present here. So I would just like to, in fact, when I went through the topic, this is really a very wide topic, I must say, life and life processes, and that too interdisciplinary approach for sustainable development. So it becomes a very wide topic for anybody to talk on. But nevertheless, of course, I shall be talking something in general about the topic of the webinar, and then something what I feel important, that is with respect to, just a minute, I'll just get this on. I have a presentation which I would like to share with you. अब इस समय उसकी जरूरत है तुम्हारे, कौन है ऐसे कंप्यूटर पे? जी जी, आप चाय पीजिए ना. So I'll just have this presentation here. Presentation share करो ना. Are the slides visible?
Not yet, ma'am.
Okay, okay. Can you see it now?
Yes, it's coming up.
Okay, okay, fine. I'm just going to is it okay?
You can just put it on slideshow, ma'am.
Yeah, it's on slideshow now.
No, it's not showing on the slideshow.
Maybe it takes a few seconds. Can you see it on slideshow?
You can just make it while presenting entire screen, then it will come.
Entire screen. Is it okay?
Yes, yes, ma'am. It's okay.
Oh, fine, fine. So now we can get along. So
Yes.
something about life and life processes, and that is interdisciplinary approach for sustainable development. So, but I think that during the past two days, you must have definitely talked about the life processes or the basic life processes. But since I have to begin here and I have to continue with what I would like to say, so these are the basic, if we talk about the basic life processes, it is the nutrition, it is metabolism, it is reproduction, it is excretion, it is respiration, it is transportation. So here all these things, they are all known as the basic essentials or the activities which are carried out by each organism so that the organism is alive. But we would say, in fact, that there are, it's more or less the similar thing, but there are 7 characteristics of life like nutrition, reproduction, excretion, growth, sensitivity, respiration, movement. And of course, if we talk about any living organism, irritability, that is the ability to respond to an environment stimulus, that is also there wherever any organism is being so now, if we want, when we talk about the life processes, it becomes also essential to correlate it with the life science because it is. This is now that science which deals with the study of all living organisms and the life processes. But I would say that it is really an enormous field of study which examines everything living which is present on the earth. It may be from the bacteria to begonias or beluga whales. Life science, in fact, it encompasses very many things. So all the life processes which we just discussed, their interrelationships, like the biology, medicine, or ecology. But if in more general terms we try to define life science, it is the sciences that have to do with organisms like plants, animals, and human beings. So when we are talking about life sciences, then of course we are moved towards biotechnology. I shall come to that later. But when we are going towards biomedical technologies, lifetime system technologies, and all different processes like environmental biomedical devices, and also how we can devote the majority of the efforts at the various stages of research, development, technology, and transfer and commercialization. So this would all, in fact, include life sciences. So how beautiful it looks when we talk about life. It varied, and just look at. I mean, if you look at the photo, nature-loving people. It may be a plant, it may be an animal. People, this is what life is. It may be an animal, it may be a plant, or even it may be just a newborn baby. And how cuddly or how do we feel when we see life? Because this is the origin of life when we talk about when a new baby is born. So all this is life. But now I see that the students who are linked up with this program, if we are going to, because the question which is in their mind is that what to do with if we study life science, what is our future, And of course, we have a large number of courses which are available under life sciences. It may be an undergrad course or it may be a postgrad course. So I'm not going to read out all these. They're in front of you, arranged alphabetically, right from agrotechnology to tissue engineering or biotechnology, which we are doing tissue mapping and all these things which have become very popular today. So coming back to the same question, what can you do with a biology degree? If we have a degree in life science, what can we do? So let us not try to read whatever has been written here. But one thing is clear, that there are many prospects. Once we do something in life science, once we study the life processes, the possibilities are many. So when we talk about a career in life science, the next question which comes into our students' mind, we have this push and pull phenomenon. In this diagram, you can see that there are different aspects in which we can be hooked up while we are studying life science, like biomedical graduates or biotech medical graduates attending medical, dental, veterinary or pharma schools, graduated but current status is unknown. See, the percentage is also given here. So now, of course, if I try to remember my own times, when I was even in my school days and I took up biology, the first thing which came into the parents' mind, because the students at that time did not think much, but the first thing that came into the parents' mind was that we have to make her or him a doctor. Biology ली है, अब इसको doctor बनाएंगे. Physics, chemistry ली है, इसको engineer बनाएंगे. So these were the two main lines anybody could think about. But today, now let us see how it has expanded. Today, when we are talking about life science, it does not restrict only to becoming a doctor. It does not restrict only to becoming a veterinary doctor. It does not restrict only to become a biotechnologist. It does not restrict only to become a teacher or a scientist in life sciences. So now here again, you can see what are the different types where we can get different careers. So this is another just representation, and the careers and job, again some more beginning again from anesthetologist. This is more, we can have all these types of careers and jobs which are linked up to life sciences. Now, suppose we take up this life science and we also get a doctorate degree. We do our undergrad, we do our postgrad, our post-graduation, and we also do our doctorate. Now we get our PhD degree. But after this PhD, the problem today in the entire country is what to do with our PhD degree. We are, and that is really, I must admit that it is a degree of a high order. So if we have this degree of high order, but with this degree, we are not going to get anything. We do not have, for example, any vacancy in the teaching job. We do not have any vacancy in to become a scientist. So then what should these postdocs do? Of course, there are certain schemes with the University Grants Commission or with the DST. They are giving postdocs like we have this DS Kothari Fellowship, many other fellowships, which are postdoc fellowships. But now, today we are with this new education policy, we are also thinking about linking them with the industry. But now industry also is not providing these positions. So now we have to look beyond the traditional career paths, which I was just discussing, and where our students, they can get along. So now these candidates, for these candidates, I know CSIR net is very important. And life science, since we are talking about the life processes or life science, right now the CSIR or UGC net, they are giving net in life science. And this problem has often arisen that we should have this net in zoology and botany separately, but till today we have this net in life science only. So these are what I have written here. These are the eligibility criteria, so they can go in for this life science net examination. And the pathway carriers, again, to list them a few: medicine or MBBS, MS or MD courses, BSc, that is undergraduate, postgraduate, MPhil and PhD courses in zoology, botany, biochemistry, microbiology. I must say these are traditional courses. Integrated PhD courses, there are integrated courses like post-graduation and PhD courses. Especially if you go to the IITs, BE, BTech and MTech, integrated MTech in biotechnology. So these courses are also coming up. So if we coming to the industry, so what are these companies which are coming up and they have become leaders? So So this just to name a few. I think I have some more here. The top biotech employers of India. So now this is, now if we have a biotechnologist, if we have our students who are coming out with a life science background or with the life processes, they can again, now Dr. Reddy's lab today, we know it has become very, very popular. Some other labs, Ranbaxy, Cisco, they're all very popular labs. So we also have certain other companies again arranged alphabetically, and where they are, you can also find them on the net. We can. What I'm trying to show is that the prospects of absorption of our students who are engaged in life sciences or in life processes, they are manifold. We can also get into forest conservation. We can also get into environmental sciences, biotechnology, bioinformatics. So all these are areas, cytotechnology. This has also become very important because cell culture, and I think most of the oncology, it is based on cytotechnology. So there it becomes very important. Medical transcription documentation, some more courses like in Vellore Christian Medical College, there are only 10 seats and the eligibility is only BSc. So we also have certain other courses like dental. Earlier we had only the dentist, that is all. But now we have, in that too, we have specializations: dental mechanic, dental hygienist, perfusion technology, which is related to cardiovascular processes, renal physiology, dialysis technology, ophthalmology and optometry. In fact, there are very many fields where we can get into. And here, this is a tree which shows you. On the left is the non-academic side. On the right is the academic side. On the center, which you see the main trunk, that is the first degree, that is a science major, master's degree, doctorate degree, postdoc. And from here, we have these offshoots, which you can see on the right and the left. We have the branches, we have the shoots, and we have the leaves, and of course, the fruits coming out when we get fruitfully absorbed in any of these branches. So now if we talk about biosciences or biomedical, it is now coming into a relatively new field of engineering, bioengineers. So till now we were talking only about biosciences, but now we are broadening our vision and we have to get into the engineering sciences as well, and we are having bioengineers. So today, just see a Microsoft engineer. He is studying the structure of the eye because he wants his computer to recognize the user by the help of his eyes. So these are some of the very modern things which are coming up, and here now comes in bioengineers, where the biological sciences and the engineers, they are merging together to give something new, something innovation. Medical instruments or artificial organs or prosthetic devices, all these things, they are all uses of biological sciences. Let me come back. So artificial, this is a special topic of interest for me, that is artificial organs. And when I used, now it's some years taking regular classes, but still I go to the postgraduate department here and take some classes here also. So I'm talking about artificial organs. This was one of my favorite topics, which I tried to teach during my immunology courses. That is how we're going for kidney transplantation. So here you can see this is a donor kidney, a very healthy kidney, and we cannot just, and on the right you can see how we transport it, because these are very delicate organs, and the transportation is also an issue. So we can transport these kidneys, and today we are talking about selective transplantation. I'm just skipping major part of this topic because it's too lengthy. It itself would take more than an hour. So when we are talking in very brief, I would say we can have whole organ transplantation or we can have selective transplantation. So often we see that we cannot get donors. So like I think you must have also noticed during this recent COVID outbreak, many of our patients died because their lung became non-functional, and we could not get donors, and the patient died still waiting for a donor. So instead, now the modern transplantation technologies, why don't we go in for selective transplantation? Instead of transplanting the whole heart, let us not simply take up real valves where the valve is not functioning. Let us take out the valve and try to transplant it. But here, these are all very technical processes. I won't go into the details, but these real valves, they are treated with glutaraldehyde at 4 degrees centigrade. And the main thing is that all the tissue, it has to be removed so that we get a bare valve. Because here, in case of transplantation, the rejection phenomena is very important. So we have to take care of this rejection phenomena, and we have to use immunosuppressants as well. If we talk about liver, we are talking about selective. I'm just focusing on selective transplantation. We can transplant only the narrow cores instead of the entire liver. In case of bone, we can use frozen and thawed pieces of bones. In case of pancreas, the functional part is the islets of Langerhans, which is producing insulin. So here we can use these islets of Langerhans, and it would be an expert, sort of technique where we are able to isolate these islets of Langerhans and give it to the recipient. But this is not a very simple. I've said it in very simple terms, but in fact it is very, very difficult, because these islets of Langerhans, they have to be really handpicked under the microscope, and it is a very tedious job. And it's isolation, because we have to enzyme digest these islets of Langerhans, so that again we do not have any unwanted tissue, because it would initiate the rejection phenomena. Similarly, now brain, I think all of us know transplantation of brain is very, very difficult. And it is difficult in the sense that, of course, we cannot take the brain of one. Brain and heart, these are two organs. We cannot take it from a person and give it to another person, because we would be depriving a life. So we take recently dead cadavers whose brain or heart are functional, and we transplant them. So, but here we are also thinking about not transplanting the entire brain, but using this pea-sized structure, that is substantia nigra, which releases the hormone dopamine, which is functionally important for us. It is helpful also in Parkinson's disease. So these are examples of how we can go in for selective transplantation. So this has also been, this is a newspaper cutting. This has always been very exciting for us, that can we have an artificial heart? We are talking about artificial limbs, we are talking about artificial eyes, we are talking about artificial organs, but what about an artificial heart? Can we make it? So Abiocord, this is the artificial heart which was made and it was being used for patients which had a non-functional heart. So here you can see this is the body of a patient, and this is a belt which charges this artificial heart, and how it has the functional properties. So that here on the left you can see this is the oxygen depleted blood which is going out to the lungs, and oxygen rich blood which is coming into the heart. So this type of system, this type of management of the heart is being done. And it was here you can see this is the Abiocord or the heart, and how it has been fitted in the human. This is the belt which is here, external battery. It has a battery. It provides the energy for the functioning of this heart. But this, it's not a lifelong type of arrangement. But of course, in cases of emergency, it can be used. The Korean scientists, they created fresh stem lines, like the donor egg nucleus was replaced by an adult cell from the same donor. The stem cells, because stem cells, these are the cells which give us the juvenile cells which can be used for further transplantation purposes. And they can be used for creating a cell line, and they can be used for therapeutic purposes also. So I think if we just try to remember, during this pandemic also, we were trying to use getting serum from donors, those who had recovered from COVID pandemic. But later on, this technique also, it was not found to be recommended for further use also. Now, uterus transplantation, this also is very important, especially for female patients where they have a hysterectomy due to hemorrhage after childbirth. So this was also a very initial step which was taken up in Saudi Arabia, so that it could make childbirth possible in women who were having hysterectomies or uterine abnormalities. Now, this is, in fact, there are 4 types of transplantation. One transplantation which we are taking from the same individual, identical twins, which is very difficult. They should be genetically identical. Not all of us have twins. Then we can have transplantation from the same person, that is from one part of the body to the other, for example, the common burns. But the allografts, which are the most common ones which we are using, that is taking grafts from one person and giving it to the other, which are specifically, they are the same, Homo sapiens, but genetically they are different. So this is the most common type where we are trying to have an acceptance rate for the transplantation. But this is the 4th type I'm talking about, xenotransplantation, because as I was telling you, often it becomes difficult to get the donors. So why not take organs from other animals? Say, for example, taken the heart or the brain from related animals like the chimpanzees or monkeys. But this is also having certain problems because people, ethically they are not tuned to it. But these experiments have been done. For example, this has been done in the United Kingdom by UKXIRA, that is the United Kingdom Xenotransplantation Interim Regulatory Authority, and this was done in this hospital. So all these experiments have been done. But there are today, why is it that transplantation is not very popular? Because first, as I was telling you, we have lack of donors. We do not get them in sufficient. Always you will find an ad that we need a donor having this blood group, because blood group is again also very important. We have to do blood grouping, we have to do tissue typing, only then we can proceed for transplantation. Then right now the cost of transplantation is also pretty high. It ranges from 1.5 to 15 lakhs. And but here in the donor, we also have a provision of paying money to the donor, like in India about $1,000 to $2,000. In Egypt, these are the figures for the different country. But nevertheless, we do have ethical problem right now. And also, I think you must have also heard about, I have deleted that part, certain scandals which had come up where this was being done in an improper manner, stealing kidneys, operating people, and then giving them to the donors, making money and all that. So I think there's no use discussing that point. But now we have to think about how we can make transplantation more successful. That is, now in this 21st century, we must, as I told you, that we have this immune response. Whereas we say that whenever we have any pathogen, we try to suppress that immune response. But now here we are giving an organ or a part of an organ from another individual or from another animal. Now here our purpose is not to suppress we want to make it acceptable. So we have to specifically control the immune response which is arising, and it leads to immunoregulation. We also talked about selective transplantation, and now for functional artificial organ transplantation, how we can make some artificial organ. I also give you a glimpse of the heart. And biotechnological advancement, this is the fifth one. It is the most recent technique where we can design or we can tailor make our organs by replacing those antigens with the donor antigens with that of the recipient. So now these are all the lines of work we can proceed. But we look forward to the day where can we have, like we go to the supermarket to buy, we say we want a Fortune brand of refined oil, or we want a particular brand of oil which you want to purchase. So can we design, or can we have these tailor-made organs or artificial hearts, livers, kidneys or lungs in the market? So I think that would take a long time, but it is an open forum for our young researchers to continue their research there. So when we are, now why don't we get donors? Often we think that when we are giving our organs, are we saving a life or are we destroying a life? So I would like to say that don't think of organ donations as giving up a part of yourself to keep a total stranger alive. It's really a total stranger giving up almost all of themselves to keep that part of you alive. It may be a kidney, it may be a simple eye, it may be a blood, it may be lymph, anything which you are short of, which is not functioning. So that stranger who has given you that, it is keeping you alive, and you're alive through that person's organ which is being given to you. So now if we are just trying to think about alternative careers after studying biotechnology of life sciences, so the Indian biotechnology industry is now coming up, and it is doing very well. But this market is very competitive, because I see many of the universities have started biotech courses. But if you look into the competitive market, they do not match, or they do not come up to that standard. So now we have to be very particular. We have to be geographically mobile. And if we look into India, it churns out thousands of biotech graduates every year. But the numbers of core domain jobs are not increasing proportionately. And that is why there becomes a stagnancy, and our biotechnologists or our life science graduates, they do not find a proper job for themselves. I'm reminded of Leeuwenhoek. Taking you back to the year 1716, my work, which I've done for a long time, was not pursued in order to gain praise I now enjoy, but chiefly from a craving after knowledge, which I noticed resides in me more than in most other men. So it is your craving for knowledge, what you want to do, and therewithal, whenever I found out anything remarkable, I have thought it my duty to put down my discovery on paper so that all ingenious people might be informed thereof. It is in very simple words, Antony van Leeuwenhoek has said that you must have a desire, a craving for knowledge. Only then you can do something new. And it is what now today we are thinking about. We are talking about innovation, how we can look into innovation. Coming back, where did we start with our research on life sciences? We started with a simple microscope. Microscope because we wanted to see some small things in a bigger size. So I'm not going into the history, but of course, when we talk about life science, I must say microscopy is an important science of investigating small objects, and that instrument which we are using is the microscope. So we have very simple microscopes which we are finding every day in every laboratory, in every classroom. But if we talk about the modern techniques or primitive, we can just see this is the evolution in the microscope, which you can see on the left, how the microscope used by Leeuwenhoek. It looks something like this. Can we call it today a microscope? Simply, he just tried to put a lens and he tried to observe it from here. This was his eyepiece and this was his objective. But then gradually we moved on, and now today we get microscopes like this with different objectives, 5x, 10x, 40x and 100x. We have eyepieces, 5x, 10x, 15x, 20x. So we can now adjust the magnification. But this too sometimes it becomes, we are not able to see our specimen properly. We have the simple microscope, we have the compound microscope, we have the phase contrast microscope, we have the dark ground microscope, fluorescent microscope, electron microscope, and many other microscopes which are coming up right now. Very good models, which we did not know about earlier. Now this, I would like to tell a little bit about electron microscopy, because I worked on this electron microscope. It was my dream to work on the electron microscope. I'll show you very briefly, because I would be failing in my duties if I do not give at least some type of technique to our young researchers who are present here, and see what is the use of this field of research in this field of life science. So this is electron microscopy is a diagnostic tool with diversified combination of techniques which offer unique possibilities to gain insights into the structure, into the topography, into the morphology, and the composition of any type of material. So this, again, just like we were talking about the simple microscope and how it looked like earlier. So this is also the primitive electron microscope. This looks something no less than a very heavyweight machinery, and a common person could not operate it. You can just see the structure. And let us now move towards now this is, I just told you about all these things. The scanning electron microscope, this is the one which I have in my laboratory. SEM, scan a gold-plated specimen to give a 3D view of the surface of an object, which is black and white. We do not have colored pictures here, but recently we are also getting some electron microscopes which are looking into this aspect also. We are studying the surface of the features of the cells and viruses, and it has a resolution. This is, here it is written 1,000 times, but the one which I have, it has 60,000 times better than the light microscope. Now, this is why an electron microscope? The question which is coming to our, which is coming to our young researchers' mind. So very broadly, I would like to say, now on the first column, we have the features of the microscope, second column, the light microscope, and the third one, electron microscope. The most important difference is that the light microscope, as the name suggests, it uses light. Electron microscopes, as the name suggests, it uses electrons. So now, not going into the very specific details, what is the resolving power, maximum magnification, here it is coming to 5 lakhs, but here, I told you it is 60,000, the one which I have. Radiation source, we are using a high voltage tungsten lamp here, and here we do not need glass lenses like we have in the light microscope. We are using vacuum. And we have the focusing screen here. We are using our common eye, but here we can see it on the screen, like you can see my presentation here on the screen. Some more features are that here we can see a live specimen under the light microscope, but under the electron microscope, no, we have to kill it. Here, the fixation is by means of alcohol. Here we are using, I used osmium tetroxide. Embedding, we do it in wax. Here, resin, in fact, we use the sticky tape there. Sectioning, we are doing microtome. This we are talking about TEM. I didn't use TEM. Stains and the grid, here we are using the glass slide and copper grid. I'll show you the copper grid. So these are the three basic different types of electron microscopes. TEM, it is just like a microtome. We are cutting the sections and we are using it in an electron microscope. SEM, I'll show you in detail, which I have done. And STEM, which is a combination of the first two. So this is, I see it in very few laboratories. So looking into the paucity of time, I'll just use a very few diagrams to explain this. So this is the electron gun which we are using here. This is the condenser lens here. The magnification control or the scan coils here. So this yellow structure which you are seeing here, this is the electron beam. When we switch on the electron microscope, we get this electron beam and it falls here on the objective lens. And this is the sample here. This is the sample which we have put here. These are the detectors here which will detect the sample, give out the electron rays here. They will be amplified here and here on the screen. You can see the display of whatever material you have there. Now this one, it's the most beautiful slide I think for understanding SEM. So now here you can see this is the electron gun, and we see step by step how our image is being formed. And this is our sample on the pink which you can see here, and we'll just create an image here. So we have the electron gun, and I'll just click here.
Sorry, madam. Sorry to disturb you.
Yeah.
We are running a little bit short of time.
Okay.
I'll
make it very brief. I'll make it very brief. So here we have the electron gun, and this is how our electron goes here, and we have here you can see the first. So this continues, and we can just have an image which is being formed here. It goes like this and this and this. I'll just skip it. And this is the microscope. You can now see this simple structure which we have here. I'm just skipping the properties. And here is this how we are taking out the sample and how we fix it here. We're taking this is isolation under stereo microscope, and this is the stud on which we are just placing our sample. This is the gold coater. We keep it here and fix it on the stud. This is the copper grid we were talking about. We just put it inside the electron microscope, press the button. This is my postdoc fellow here, and here we have the image which is being formed here. This is the entire setup. And just some pictures on the left and right. This is SEM, and this is the light microscope. I'll just go through these pictures. You can just see, this is, if you remember, these are the pictures of, if you remember the liver fluke, can you just imagine these? This is a ventral sucker. This is simply the margin of the ventral sucker, which you can see finger-like projections. And not going into the details of the structure. So this is a nematode parasite. These are the sensory organs which you can see. This is again a nematode parasite. You can see the wonderful lip structure. Eggs which we could accidentally, we could make the SEM preparations. Tinea, I think you all see, but this is from the rat. You can see the beautiful basket-like structure here, and the uses of electron microscope. There are very many today. In fact, it is coming into industry. It is going into very many different fields. That's finishing up. So we have to design these materials. We have to look into the possibility of how we can use nanotechnology. This has become the future of science today. And just skipping where we are going today, we have newer microscopes and how we can use our skills in problem solving. And the trained scientific innovators, we have to have a mastery in any field of life science at the international level only, and we are moving towards innovation when we try to do something new in life science. These are simply the lists of the different institutes and the job profiles, and I'm sure that we are definitely, this webinar is going to give us something new on life sciences and life processes, and I'm sure that this webinar is going to be successful. And I'm sure that we shall get some very good conclusions and recommendations which can be taken up through the proceedings of this webinar. I once again thank you very much for giving me an opportunity to present some of my views here on life and life processes. Thank you so much.
Thank you, ma'am, for your insightful talk about the opportunities in life sciences and more specifically in zoology, and also updating us about the latest trends in science and technology. I'm sure the participants, particularly the PhD and MSc students, are now aware all of these potential opportunities and also the scope of applied zoology. Once again, on behalf of the organizing committee of this seminar, I offer my sincere gratitude to you, Honorable Ma'am, the Vice Chancellor of Tilka Manji Bhagalpur University, Professor Neelima Gupta Ma'am, for taking the time out from your busy schedule to deliver your address. Once again, I humbly thank you. Thank you.
Yeah, thank you. Thank you.
On behalf of Department of Zoology, I thanks to Dr. Neelima Gupta, who was a zoologist once upon a time, and now administrator as academician head in the universities. So it is very tough to get a time to give a talk in the seminar of this type of platform. Still, you managed to give some time. We are greatly grateful to you. Thank you, madam.
Thank you. Thank you. Thank you.
Thank you, sir. We will now move on to the last invited talk of this session. That is of Dr. Shobna Kapoor. So right now I would like to introduce Dr. Shobna Kapoor. She is a professor at the Department of Chemistry in IIT Bombay, Mumbai. And she is a biophysical chemist and chemical biologist working at the interface of physical and life sciences. She obtained her PhD in biophysics from Technische Universität Dortmund, Germany, and carried out her postdoctoral research in chemical biology at Max Planck Institute of Molecular Physiology, Germany. She started her independent career at IIT Bombay in December 2016, where her group is interested in the multiple roles played by lipids in infectious diseases at the molecular level. She is a recipient of the DST Inspire Faculty Award in 2017, INSA Young Scientist Medal 2020, NASI Young Scientist Platinum Jubilee Award 2020, SERB Women in Excellence Award 2021, and Indian Alliance Wellcome Trust Intermediate Fellowship. With such an illustrious career, on behalf of the organizing committee of the National Seminars of Life and Life Processes, I welcome Dr. Shobna Kapoor and humbly request her to deliver her talk. Ma'am?
Yeah, thank you, Dr. DeCosta, for a very nice introduction. First of all, I would like to have this lively seminar.
Ma'am, we are. Can you be a bit louder? We are unable. Yes.
Can you hear me now? Yeah.
Yes.
Okay.
Yes.
Yes. So I would like to thank all the organizers of this wonderful meeting, Professor Roy, Dr. Desai, and Dr. DeCosta, for giving me an opportunity to share my results. So I will just give a glimpse of the work that we have been doing in my group. Yes. So I hope everything is it visible? My screen?
Yes.
Perfect. So this is the last talk, technical talk in this session. And I think so my life is a little bit easier because Professor Roy already gave a very nice introduction on the importance of lipids, in mitigating, or the potential role in mitigating various kinds of diseases like hepatitis, alcohol damage, and even diabetes. Whereas what I'm going to discuss in the couple of next 15-20 minutes is we are working with lipids from infectious agent, which is Mycobacterium tuberculosis. And I would like to basically showcase that how these lipids are involved in progression of the infection, which up till now has been largely dominated by proteins. Before that, as I said, my group is a relatively new group where we work with different aspects of lipids. And what I'll be talking about is basically the mechanism of pathogenic lipids in diseases. So as I said, that we work with pathogenic lipids from Mycobacterium tuberculosis. On the right-hand side, what you can see is a unique cell wall lipid structure of Mycobacterium tuberculosis, again, which does not need any introduction. It's an agent that causes the highly infectious disease TB, tuberculosis. And if you look at their cell wall or lipid structure, it's highly complex. For example, mammalian cells that we deal with have only a plasma membrane. But these organisms, they have a very thick cell wall, and the lipids that we are interested in are the ones which are present non-covalently bound in the outermost layer. Why this is so is because there has been, in the last few years, there has been some reports which have actually stated that in addition to proteins, these lipids can also be secreted by this agent during infection in the host cell, which is our cells, but their molecular mechanisms have not been elucidated. So coming from a lipid chemistry background, this topic caught my interest, and I established my group to work at that particular interface. And the main question that we wanted to ask is how the infectious lipids present in this bacteria, they alter the host cell membrane properties. So first we designed an infection model wherein we purify the lipids of mycobacteria. So just to put things into perspective, this is the structure of one of the pathogenic lipid, which is tetraisylated because it has 4 chains, highly branched with methyl branches, having a trihalose sugar as well as a sulfate group. Therefore, it is called as a sulfoglycolipid. What we did was we basically took mammalian cells. In our case, we took THP1 macrophages, which is a very commonplace model cell line used for infectious diseases work. And we followed the insertion of these exogenous bacterial lipids in the cell membrane of these host cells and asked the following questions: that what changes do we bring about in membrane organization of the host? Does it modulate membrane dynamics? What about downstream processes like signaling, etc.? So the first thing, of course, what we had to do, we had to verify that we indeed have these exogenous lipid inside the host cell. So we did that both by using TLC extraction as well as by mass analysis. So what you see here is that these middle TLC lane is for a controlled purified SL1 lipid, where you see a band here. The first row is the total lipid extracted from just the mammalian cells, where you do not see any signature of this exogenous bacterial lipid. And in the third lane, you see, in addition to the mammalian lipids, you also see a band of these exogenously added lipids, indeed confirming that once we incubate these mammalian cells with suspensions of these bacterial lipids, they insert in the plasma membrane of these live cells. The next method that we used to study basically the membrane domains or dynamics was 2-photon microscopy using Laudan. Now, this is the structure of Laudan, which you can see on the screen. It's a solvochromic dye. It's a fluorescent dye. So what I mean solvochromic dye is that if you look at its emission, fluorescence emission properties, it changes depending upon the microenvironment around the dye. So when the dye is in a very tightly packed lipid bilayer, it emits at a lower wavelength. When it is in a very fluid or disordered bilayer, it emits at a higher wavelength. So using a ratio of the intensity of these two wavelengths, you can actually calculate a polarization factor, which is known as generalized polarization, or GP, which you will often hear the term in my presentation. And in layman words, what GP basically means, if you have a very high value of GP, positive value of GP, which is the in the red color bar here, it means that the membrane is highly packed. If you have very low GP, that means towards the blue end of the spectrum, that means your membranes are relatively hydrated or fluid or disordered. So we injected we incubated this dye with our live THP1 cells, and what you see is not a conventional fluorescent microscope image, but rather a 2-photon GP map, where each and every pixel gives you a value of this parameter GP. So outrightly, what you can see is that the surface of mammalian cells, the lipid membrane of these mammalian cells, it's not homogeneous. Actually, it's very heterogeneous with respect to the lipid environment. And you can see these orange colored dot-like structures, which basically suggest that there are certain domains on the surface of these mammalian cells, which are very tightly packed, as shown in this cartoon representation here. Fine. But what exactly presence of these domains, mean? So this means that you have lipid nanoclustering on the surface of mammalian cells, and indeed it has been shown as a functional feature of plasma membrane of various cells. And when I mean by nanoclustering means that if you take, let's say, a 3D snapshot of a super resolution image of a plasma membrane of a mammalian cell, you will see that there are hills and valleys on the surface of these cells, which are actually the domains that the lipids form. That is basically domains within the lipid bilayer. And why they are important is because these domains, as you can see in the blue color representation here, these domains basically act as hotspots for recruiting proteins, for initiating signal transduction processes. And the logic that we had was that any agent that can kind of disorganize this kind of a lipid domains will have implications of cellular function. So therefore, the questions that we wanted to know is whether our mycobacterial glycolipids can alter this domain organization of live mammalian cells after infection. And what we did saw was indeed yes. If you compare the GP graph of a control cell with the one which is treated with the glycolipid at a given condition of 4 hours and physiological relevant concentration, you see that the surface morphology of the cell membrane has changed. Instead of seeing these dot-like orange structure, what you see is sunflower-like pattern, suggesting that there is some kind of reorganization of the lipid membrane on these live cells by this lipid. We are still trying to understand what are the molecular determinants of this reorganization, but indeed insertion of these infectious lipids induces a change in the global organization of the plasma membrane of these infected macrophage cell lines. We were able to not only monitor that qualitatively, but also quantitate the degree of changes by calculating a histogram of the GP and calculating, deconvoluting these distributions into Gaussian distribution. We were able to calculate the area of your high GP domains, which is basically the domains on the live cell membranes, which are very high GP, or you can say that they are highly ordered lipid domains, which decreases. The next thing is that the results that I showed you up till now were of the global cell membranes. And in order to just look at specifically the plasma membrane, we use different kinds of tools, image segmentation tools. And we were able to actually see a similar effect on the plasma membrane compared to the global changes in the lipid membranes. And that is this is because both the dye as well as the mycobacterial lipid that we use can internalize into internal cellular membranes as well. So we wanted to see whether the effect is limited only to the plasma membrane, or do we also see effect in other internal cellular membranes. And we were able to see that there is an effect on both plasma and internal membranes. The next thing that my group decided to do was that in addition to the sulfoglycolipid, which is shown here, we looked at other structurally diverse lipids. And as you can see, their structures, they are very huge compared to mammalian lipids. Some of these lipids have chain lengths of carbon 90 compared to carbon 20, which is found in the standard phospholipids. So we decided to probe, all these lipids together. And what we found was that very interestingly, the structurally different lipids of MTB, they actually have a different effect on the lipid domains of host cells, suggesting that the microorganism or the bacteria, it can use different lipids for inducing different extent of perturbation on your host cells. Of course, the biological implication of this still has to be completely studied and verified. So after knowing that our lipid domains were getting reorganized or redistributed, we wanted to probe further. What about membrane dynamics? Does the dynamics also affect it? So for that, what we did was we took two different probes. The first probe, which is shown on the top left-hand side, is basically the one that partitions in the ordered regions of your lipid membrane because it has all saturated chains. And the lower right dye has a double bond, as a result of which it can partition in the disordered regions of the plasma membrane. And you can see both in the heat map as well as in the normal confocal microscope images that the dye has gone in the plasma membrane of these THP1 cells. So what we did was, compared to the control, we monitored the diffusion of both the dyes as a function of different lipid contributions or insertion into their cell membrane. And what we found was if we looked at the diffusion coefficients as well as the mobile fraction, which we obtained from the fluorescence recovery after photobleaching experiment, that different lipids have different effects. For example, whereas the sulfoglycolipid was able to increase diffusion in the fluid region, the trehalose diamycolate, which is another structurally unrelated lipid, was able to decrease the diffusion. So clearly, different lipids tend to have different effects on the diffusion of plasma membrane components. The third thing that we wanted to explore was what about membrane mechanics? Does it affect membrane stiffness or elasticity? And that is because usually it has been reported that pathogens, they modulate the host cell stiffness or elasticity in order to foster their uptake. So we decided to look at the specific contribution of these lipids in mediating this process. For this, we used AFM, atomic force microscopy. And what you see in a control representation here is that if you take an AFM cantilever tip and you try to come near to the surface of the cell, this is the nucleus, this is actin cytoskeleton, and this is the cell shape. When you start to indent and retract, what you can do is you can monitor these kind of curves, which is force versus the distance during the indentation and during the retraction. And from these curves, you can fit it to different kinds of models and calculate your elastic modulus. And if you plot the distribution of elastic modulus for both the control and treated cells, we found that there was a decrease in the stiffness, which is in alignment with the fact that mycobacteria in general as a whole organism can induce softening of the host cells to foster its uptake. Now we know that one of the component of the bacteria that is mediating this effect is its unusual lipids. The next variant of AFM that we used was tether microscopy. Now, what are membrane tethers? They are thin nanotubes that regulate cellular communication, adhesion, and immune responses. And you can see them in this cartoon representation here. So when you have the AFM cantilever tip indenting, which is here, and when you retract the tip, sometimes these thin membrane nanotubes, they stick to the surface or tip of this cantilever. And by measuring the force with which you are, retracting it back, you can observe these kind of tether let's say breakage points, which you can monitor and quantify. So for example, here in this cell, you have 3 tethers, then 2 tethers and 1 tether, and each of this is represented by, each of the tether is represented by a region of constant force or a plateau or constant force region in a force versus distance curve. So you can quantify the number of tethers and you can quantify their length as well as the force with which you tend to break the tethers, and that gives you information about membrane stiffness as well as resistance to bending, which are all very important if you consider membrane remodeling during phagocytosis or endocytosis, which are all modulated by the pathogen during infection. And what we found was that the sulfoglycolipid 1 treated membranes required less force to be extended because their surface tension or bending rigidity was smaller. The number of tethers per cell increased, but that length was found to decrease, suggesting that the total membrane reservoir is not altered by these lipids, it's only redistributed. So with this, what we did was we extended the study to other lipids, as I have already shown, to other structurally distinct lipids, and we found, without going into the detail of this data, what basically we found is that different lipids of the mycobacteria show differential effect on membrane stiffness, fluidity, as well as organization dictated by the nature of these lipids. For example, whereas the saturated chains in this PDIM tends to increase the stiffness, whereas the methylated branches in SL1 tend to decrease the stiffness and hence impact the fluidity and diffusion of plasma membrane components in a different fashion. The next thing that we were interested is whether the effects of these mycobacterial lipids are only restricted to the plasma membrane properties or may also percolate to the actin cytoskeleton, which is underneath the plasma membrane. And this is because actin polymerization or depolymerization provides a lot of mechanical forces for processes associated with cell shape changes, migration, and we know that the pathogen in general can modulate actin network to foster its infection and progression as well as survival. So we followed the actin morphology in control as well as in treated cells, live cells with different treated with different lipids, and we were able to quantify the total abundance of actin cytoskeleton, which seemed to be altered differentially by different lipids. Some of the lipids had no effect, whereas some of the lipids increased the total actin volume. And going deeper into this data, we tried to look at actin not only in terms of just bulk actin, but in terms of filaments, patches, or puncta. And we found that different lipids actually regulate or redistribute actin in different morphologies. And this study, for the first time, actually revealed that mycobacterial lipids are a key mediator of this actin redistribution, which had been observed in early 1970s using the intact bacteria. So one of the last question that we wanted to look at is that, okay, we know that MTB lipids differ in their structure, and that affords selectivity in modulating membrane dynamics and actin. But does a single lipid itself has additional modules that can regulate selectivity? And here our expertise of chemical biology comes in, where we made a synthetic analog of the sulfur-glycolipid. As you can see here, compared to the natural lipid, it had truncated palmityl chains at the 4 positions on the trehalose head group, as well as a sulfate, and we created different analogs. For example, analog of sulfur-glycolipid without a sulfate, so that we can see what role does the sulfate group has to play in this lipid structure. When we remove 2 chains at the 6,6 prime position or at the 2,2 prime position of the sugar group, how does it impact? So without going into details of these experiments, what we were able to show using a combination of biophysical and cell biology experiments was that the sulfate ion is very critical for membrane hydration of the host cell, whereas acyl chains, which are positioned at the 6,6 prime or the 2,2,3 position, have actually opposite effects on membrane microviscosity, or in other words, membrane fluidity of the host cell. And this basically led us to conclude that specific chemical moieties within the single MTB lipid structure, be it its chain, branches, sulfate, position of the lipid chains, they can act as regulators of fine-tuning the host lipid interactions, which can have very important implication during the progression of the infection. And we verified these results using molecular simulations, where we simulated the sulfur-glycolipid 1. And what we were able to see is we were able to monitor the order of the, of the 3 carbon chains, the long carbon chains of the sulfur-glycolipid. We were able to monitor its conformational order throughout the length of these chains. And we were able to see that they indeed form bilayers which are very heterogeneous. We were able to confirm it experimentally. And what you see here is a confocal microscope of a giant unilamellar vesicle, which is made of a single component lipid, and yet it is able to show phase coexistence with the red regions which are accumulated with the fluorescent dye, whereas the flower-like patterns that you see are basically the membrane regions which are devoid of any dye. So a single component lipid from mycobacteria is capable of forming this phase segregated system, which is a field in itself which we are trying to explore further. And what we were able to see is that the sulfur-glycolipid 1 actually can exist in 2 different phases, one which is very flexible or has a high flexibility in its chain, and the other which is more extended. And that could give a reason of why the single component system can phase segregate, and also explain to some extent the different biological results that we have obtained. So with this, I think so I would like to conclude and put all of the things together, that using different structural they can insert in the plasma membrane of the host cells. They can alter your membrane organization. They can alter the membrane mechanics as well as the diffusion of the plasma membrane. we could see a disruption of the actin cytoskeleton. And what I did not show you today was basically we monitored the functional outcome of these interaction by monitoring the cytokine production and autophagy, as well as various biochemical processes like protein phosphorylation and lipid peroxidation, wherein we saw a lipid-dependent effect on these processes. So we propose at this point that there might be a previously unknown mechanism by which this microorganism can fine-tune its interaction with the host cells, governed by modulating the lipid profile on its surface. So for example, when it wants the cells, host cells to be stiff, it can increase the production of PDIM on its surface. If it wants the host cells to be less stiff so that it can foster its uptake, it might increase the production of SL1 on its surface. Of course, this is the hypothesis that needs to be tested, and that the unique conformational and structural makeup of these lipids actually regulate lipid membrane structure and function to great extents which previously remained unknown. And with this, I would like to acknowledge my group, for sharing the same vision as me, my collaborators, the Department of Chemistry, IIT Bombay, my funding sources, and again, thank you all for your kind attention.
Thank you, ma'am. Thank you, ma'am. Thank you, ma'am, for that insightful talk on Mycobacterium tuberculosis. Thank you, ma'am, for that insightful talk glycolipids, Mycobacterium and postal membrane and three-wire membrane associated functions during infection. This was a very insightful talk where ma'am spoke about the role of sulfoglycol in these molecules. They play a very important. They, in fact, are very critical in membrane associated. Also, she spoke about the different factors which can also affect these processes. Again, ma'am, on behalf of the organizing team of this national seminar, I extend my thanks to you for this wonderful presentation, and I'm sure the participants have definitely gained a lot of information with regard to your research and also the potential opportunities that are available in lipid biochemistry for them as well. So once again, I thank you, ma'am. We will now move on to the last section of this seminar. So after a fruitful 3 days of really technical sessions, wonderful talks, presentations, and posters, we now come to the end of this seminar. So we will now commence the valedictory session, and I would request the convener of this seminar, Professor Roy, to welcome the gathering.
A very good afternoon to all of you. Dean Professor P.K. Sharma, Faculty of Life Science and Environment, former Head of the Department, Dr. A.B. Shanbag, as a guest of honor, distinguished speaker, delegates, students, and my colleague. We are at the end of 3-day national seminar on life and life processes, interdisciplinary approaches for sustainable development. The success we achieved to organize this national seminar is possible as we received the delegates and participants from 18 states of our country, including the extreme north like Jammu and Kashmir to south, Kerala and Puducherry, extreme east, that is Kolkata, to extreme west, that is the Gujarat. So the success we achieved for this event is also due to the contribution of each and every member of the family of zoology department. We consider every stakeholder of our department as a member of our family. It includes the student, their parents, supporting staff, and the faculty member. As I mentioned earlier, I am the last person among the faculty members who joined the department in the first 2 to 3 years of its establishment. I feel honored myself with the recent recruitments of all the young faculties who were once upon a time were my student, and now I welcome them as my younger colleague. I am confident that they will collectively take the department to a greater height in academics and strengthen the bonds of the zoology family. As I mentioned earlier also, from the last academic year, we started 3 cluster optional courses, namely aquaculture, life processes, and wildlife biology, each of 12 credits as a steps towards the development of skill among the students. These cluster courses is having 1 credit internships. We hope that each of the cluster optional courses will facilitate the student mobility across the different institutions within the country and abroad. And also they'll be potent employer in the pharma industry, biotech industries, and medical biotech diagnostic industry. Besides also, this cluster will also help them to start the self-entrepreneurship program like ecotourism, like development of the fish farm or farm management, etc. Therefore, before superannuating, using this platform, I appeal to all present-day stalwarts in zoology. It may be the court zoologist, maybe the modern biologist or the aquaculture biologist, to extend their hand of cooperations to my younger colleagues as and when required. And your cooperation is essential to achieve the success in order to transmission of the knowledge among the youth generation of Goa and help them to develop some skill among them. With this, I will conclude my speech. Once again, I thank each of you for your wholehearted support for this 3-day national event. Thank you.
Thank you, sir, for that welcome note. We have amongst us the chief guest, Professor P.K. Sharma, and guest of honor, Dr. A.B. Shanbag, for this valedictory session. I would now like to introduce our chief guest, that is Professor Prabhat Sharma. So it is an honor to introduce him at this valedictory session. Professor P.K. Sharma is the professor in the Department of Zoology and is the Dean of the Faculty of Life Sciences and Environment at Goa University. He has completed his PhD from King's College London in 1990, and he has extensive postdoctoral experience such as in the Imperial College London from 1989 to 1990 and 2007, Rothamsted Agriculture Research Station, Harpenden in UK from 96 to 97, University of Cambridge in UK in 2006, and the University of Exeter in UK from 2007 to 2008. He is the recipient of several awards such as the Young Scientist Research Award in Food and Agricultural Sciences, which is bestowed under the age of 35 years, Department of Atomic Energy, Government of India, 1994, Professor Hiralal Chakravarti Award, and a citation from the Indian Science Congress Association in 2002, DBT Overseas Associateship by DBT Government of India, April 2007 to July 2007, INSA Royal Society Fellowship 2006, and Commonwealth Academic Staff Fellowship in 1996. Respected Sir has completed 15 projects from various funding agencies, including being a coordinator of UGC SAP from 2012 to 2020, and deputy coordinator from 2004 to 2012, and DST FIST 2010 to 2015. He has a large number of research papers in established international and national Scopus journals with a cumulative impact factor of more than 50. On behalf of the organizing committee, I humbly welcome respected Professor Prabhat Sharma to this valedictory program. It gives me immense pleasure to introduce the guest of honor of this valedictory session, Dr. A.B. Shanbag. Dr. A.B. Shanbag is the ex-head and professor of the Department of Zoology, Goa University during 1992 to 2013 with 21 years of service. He has completed his PhD from Karnataka University, Dharwad, Karnataka, and he has also completed postdoctoral training in immunocytochemical studies on hypothalamus in Roslin Institute, Edinburgh in UK. Back in India, he worked on field biology with emphasis on bird ecology, monoculture, forest ecology, and wetland biology. His services were also utilized by the government of Goa as a member on the task force on Karambolim Lake development and also the State Wildlife Advisory Board. He is also on the Research Monitoring and Advisory Committee of Salim Ali Center for Ornithology and Natural History, SACON, in Coimbatore, Center of Excellence of Department of Environment, Forests and Climate Change, Government of India. He has published several research papers in peer-reviewed national and international journals, besides full-length reviews and chapters in edited books. He has successfully guided 4 research students towards their doctorate degrees. And on behalf of the organizing committee, I humbly welcome respected Dr. A.B. Shanbag to this valedictory program. We now recognize the services of our former HODs in a special felicitation function, which will be bestowed upon by Professor R. Roy, that is the head of the Department of Zoology, Goa University.
Sri M.P. Tonsekar, ex-faculty of Chogle College, served the department as coordinator on deputation when the department started in the year 1991. Sri Tonsekar, along with the help of the contributory lecturers from undergraduate colleges affiliated to Goa University, managed the teaching curriculum of the department in the beginning 1 or 2 years. His support as coordinator will be always remembered by the Department of Zoology, Goa University. So, since he is not there, he is very aged person, I request our organizing secretary, Dr. Shanti Desai, to convey this felicitation certificate to him at his residential address, Margao.
Yes, sir. Yes, sir. Thank you. Thank you.
Dr. S.Y. Paranjape joined the Department of Zoology, Goa University in 1994 as a professor, and he was there till 1996. He introduced the entomology to our curriculum and provided a wide range of the tools that's required for to study the entomology research. And still we are using those tools for our collection of the insects and pests and identification of them. It was a pleasure to have his service for the Department of Zoology, although it was a very short duration. Again, Professor Paranjape is stationed at Pune. I think he couldn't able to join the online meeting. Once again, I request the organizing secretary, Dr. Shanti Desai, to convey this felicitation certificate to him at his Pune address.
Yes, sir. Thank you.
Is there P.V. Desai? Dr. P.V. Desai joined the department in 1993 as a reader and superannuated in 2012 as a professor and head of the Department of Zoology. His research specialization is in the area of physiology and more particularly about the neurophysiology. He has several publications in reputed national and international peer-reviewed journals. He guided several PhD students. As a member of several committees of the university's bodies, he served the institution besides his main role as a teacher and researcher. Dr. P.V. Desai was served the department faculty as a Dean of the Faculty of Life Science and Environment. He also officiated the university as a capability of the Registrar for some time. So we are at the Department of Zoology of Goa University are deeply in depth to Dr. P.V. Desai for his outstanding contribution in the department as well as in the university. Again, due to bad connectivity, Dr. P.V. Desai, he was there our inaugural program and delivered the guest of honor address. But today, due to the bad weather at Maharashtra, he couldn't able to connect us. Again, I request organizing secretary, Dr. Shanti Desai, to convey our felicitation to him at his Maharashtra, Mumbai address.
Yes, sir. Thank you.
Dr. A.V. Shanbhag joined in the department in 1992 as reader and superannuated in 2013 as professor. He was the first person who took the responsibility of the department to lead as a head in starting from 1992. And obviously the headship, our rotation, okay, but he has served 2-3 times as a head of the department. His research specialization is in the area of avian biology and wetland ecology. He has also several publications in the reputed international national peer-reviewed journals, guided several PhD students. As a member of several committees in the university's bodies, he served the institutions beside his main role as a teacher and researchers. We at the Department of Zoology, Goa University are deeply in debt to Dr. A.V. Shanbhag for his outstanding contributions in the department as well as in the university. I request Dr. Shanbhag to accept these felicitations from the department. Dr. Shanbhag, please respond.
Yes, thank you, Professor Roy. I am very much grateful and obliged to the department for bestowing me with the certificate. Thank you.
Dr. S.K. Sharma joined the department in 1992 as a lecturer and superannuated in 2013 as a professor and head. His research specializations is in the area of genotoxicology and human genetics. He has several publications in his reputations in various international and national peer-reviewed journals, guided several PhD students. He also given his laboratory as a platform to pursue the postdoctoral students. As a member of several committees of the universities, beside served as the institutions, beside his role as a teacher and researchers. We at the Department of Zoology, Goa University are deeply in debt to Dr. S.K. Sharma for his outstanding contributions in the department as well as in the university. Again, I request Dr. Sharma to accept our felicitations. Dr. Sharma?
Yeah. Can you hear me?
Yes.
Thank you, Professor Roy, and the whole departmental council for the felicitation function and felicitating me. And, there's a time I want me to tell something.
You can tell 1 or 2 words, no problem.
So I wish to congratulate the department in general and the organizers of this event in particular for your successful conduct of this national seminar, the special session in the honor of Professor Roy on the occasion of the superannuation of the last of the department's founding teachers, the first generation of the teachers of the department. This will be a great occasion of the initiation of the process of handing over of the department by the last of the first generation teachers, Professor Roy, to the next generation of newly appointed bunch of our talented young teachers. I wish to encash this opportunity of my felicitation to thank you all the individuals associated with my long career of around 3 decades in Goa University. I'm grateful to each and every one of you, all associated with me, who have contributed either directly or indirectly, including the whole community of students, teacher, colleagues, and the administrative staff of Goa University, ranging from the vice chancellors to the registrars, deans, HODs, and even up to peons. Okay. I don't want to take any names here because the list is infinite. All have contributed immensely towards the smooth sailing of my tenure as a teacher in the department in Goa University. I could achieve my present status because of the kind help and cooperation extended by all of you, and hence at this moment I humbly wish to express my sincere thanks to each and every one of you for whatever help you rendered uh, and extended to me in the Department of Zoology of Goa University. Further, my best wishes are to all the delegates of this seminar for their success in the future. And a piece of appreciation and I also wish to pray and beg the Almighty to guide and bless you all, our successors, uh, that is the present generation of next generation group of young teachers of the Department of Zoology of Goa University to serve mankind to their full capacity and thereby to take the name and fame of the department and also of Goa University to greater heights of recognition at the national and international level in future. Best wishes to all of you. Thank you.
Thank you. Change? It's not change.
There's a small technical issue. We'll get back.
Just come to start. It doesn't look. Let it should be shown first. Sorry for the technical disturbance. I'll continue. Dr. I.K. Pai joined the Goa University Department of Zoology in 1991 as a lecturer, and in 2019 he superannuated as a professor and head from the department. Dr. Pai's research specialization is in the area of the various aspects of the biodiversity. He has several publications in his reputations in national and international journals, guided several PhD students. He also served as a member of several committees of the university bodies, and he also served as a director of the Academic Staff College, which is known as the UGC HRDC Goa for a few years. So besides his teaching and researcher role as a teacher and researchers, he contributed in various administrative aspects of the university. We at the Department of Zoology, Goa University, are deeply indebted to Dr. Pai for his outstanding contributions in the department as well as in the universities. So again, I request Dr. I.K. Pai to accept the to accept the felicitations. Dr. I.K. Pai, please accept our felicitations.
Thank you very much, Professor Roy and his team, for thinking of this wonderful program of remembering the superannuated teachers. This is happening for the first time in the history of 31 years of Goa University Zoology Department history. I think I consider this as a good practice what Professor Roy and his team has started. I'm sure this practice will continue in future also to remember the persons who have contributed for the department, its welfare and the betterment of the department. Thank you very much. Wish you all the best for all of you. Have a nice day. Thank you.
On behalf of my personal and Department of Zoology, once again, I show my gratitude to all the ex-colleagues of my department with whom I work around almost 25 to 28 years in the department with a lot of ups and downs. But at the end, we are the one family, that is the zoology family. And that message needs to go in future and coming days. Thank you.
Thank you, sir, for that heartfelt gesture. That was from the Department of Zoology, Goa University. I now request Dr. Meenal Shirodkar to present the report of the national seminar.
Good evening, one and all. The 3 days national seminar on life and life processes, integrated approach for sustainable development commenced on 19th July 2021. The chief guest for the inaugural function was Professor Varun Sahani, Vice Chancellor of Goa University. Professor P.V. Desai, former Dean of Life Science and Head of the Department of Zoology, was the guest of honor. There were 4 sessions in this seminar. The first session was on life and life processes, economic empowerment, life forms, habit and habitat, integrated approaches in life processes, well-being state, a sustainable step towards lipid biochemistry. We had 4 plenary talks by eminent researchers from esteemed institutes. Plenary talk for the second session was by Dr. Kailash Chandra, former Director, Zoological Survey of India, West Bengal, and his talk was on life forms, habit and habitat. He gave an elaborate insight on biodiversity wealth of India, of different ecosystems of India. His talk ranged from emphasis on faunal database of hotspots to DNA barcoding of several species. Plenary talk for the session 3 was by Professor Arun Pandopadhyay, Director of Indian Institute of Chemical Biology, Kolkata. He spoke on plasma proteomics, reveal dysregulated cholesterol transport and compromise inflammation resolution in coronary artery disease. Another plenary talk in this session was by Professor Prakash Babu, Dean, School of Medical Sciences, University of Hyderabad, Telangana. He enlightened us with his talk on translational research for treatment of brain tumors. In session 4, we had a talk by Professor Redanna Pallu, Professor, University of Hyderabad, Telangana. His talk was on eicosanoids, the bioactive lipids in the regulation of cellular homeostasis. We had 9 invited speakers from well-known institutes of India. One of them were Professor Sumit Homi Chaudhary, Head of the Department, Zoology, University, Calcutta. Sir briefed us about the emergent properties in hierarchies and productive value for ecological shifts, ecosystem stability, riverine ecosystem, and ecological fish printing, which is a relatively modern accounting tool that can be used to help distinguish between sustainable and unsustainable aquaculture practices. Dr. Shrikant GB, scientist from ICAR, Central Coastal Agricultural Research Institute, Goa, spoke about fisheries sector of Goa, present status, challenges, and opportunities. In session 2, the invited talk was by Dr. Suresh Kumar. He delivered a talk on population status and conservation of a top predator of aquatic ecosystem, the spot-billed pelicans of peninsular India. He gave a glimpse of waterbirds in general, elucidated on heronries in India, and emphasized on heronry in Karnataka. Dr. Dattesh Desai, scientist from National Institute of Oceanography, Goa, spoke on the topic entitled Implications of Habitat Characteristics in Structuring Benthic Life. He stressed on ecology of benthic fauna and Zuari estuary of Goa. Dr. Ashwini Kumar, Director, Indian Council Malaria Research, Vector Control Research Center Medical Complex, Puducherry, spoke on One Health approach for human well-being and sustainable development from a concept of reality. Dr. Manjula Das, senior researcher from Muzumdar Shaw Center of Translational Research Institute, delivered a talk on a novel recombinant protein to fight COVID-19. Professor Subhash Peter, professor from University of Kerala, spoke on understanding life processes, lesson from integrative biology. Dr. Anshuman Sarkar, associate professor from Biology Department of BITS Pilani, Goa campus, talked about old wine in new bottle, exploring the potency of anti-malarial drug from treating breast cancer. Professor Shobna Kapoor, Professor, Department of Chemistry, IIT Mumbai, delivered a talk on Mycobacterium tuberculosis, glycolipids disorganize, host cells, membranes, and rewire membrane-associated function during infection. We had a most interesting and enlightening concluding talk by Honorable Vice Chancellor, Professor Neelima Gupta. For this national seminar, we had 84 delegates from 18 states registered. There was wide coverage of participants from Jammu Kashmir to Tamil Nadu and from Gujarat to Assam. There were 19 oral presentations by faculty members from colleges and universities from all over India. 38 poster presentations were done by students pursuing PhD, and around 40 of our MSc students attended this conference. So this was a brief report of 3 days national seminar on life and life processes, integrated approach for sustainable development. Thank you.
Thank you, Dr. Minu, for presenting the report of the national seminar. We now move on to the moment that we have all been waiting for, and that is the winners of the poster presentation. So I humbly request the guest of honor, Dr. A.B. Shanbag, to announce the prize winners of the poster competition.
So please unmute yourself.
Am I audible now?
Yes, sir.
It is a privilege given to me for announcing the prizes for poster presentation. First place goes to Navdeepa Bhattacharya from BITS Pilani, Goa campus, for presenting the work on anti-cancer properties of 2 small molecular weight toxins from Russell's viper venom. And second place goes to Arghya Laha from University of Burdwan, West Bengal, for presenting her work on role of HLA-DQ and IL-13 gene polymorphism in shrimp allergy among West Bengal population, India. Thank you.
Thank you, sir, for announcing the prize winners. I now request Sir Shanbag to address the gathering as the guest of honor.
Good evening, everybody. Professor Sharma, the Dean of Faculty of Sciences and Environment, Professor Ramballav Roy, the head of the Department of Zoology and also the convener of this particular symposium. I'm sorry, it is the virtual conference by the department, virtual national conference. And Mrs. Chanti Desai, who happens to be the organizing secretary for this particular conference, and all the members of the faculty of the zoology department. I may not be wrong in if I refer the rest of the members of the zoology department as my students, because almost all of them are either my students in the graduation and post-graduation sometimes only in post-graduate department, or sometimes might be students of my students, that is to say grad students. So in that respect, I am justified in referring to the next batch of faculty in the Department of Zoology at Goa University as my students. Now, a couple of months back, when I was in contact with Professor Roy, he sent me a flyer of this particular conference, and I was very much elated by looking at that flyer. It was colorful, it was well represented as to the overall biodiversity, the various aspects of zoology, and so on and so forth. So I sent a congratulatory message to him. He wrote me back telling that it is the probably the last conference of his active professional life in the department. At that point of time, I knew that this particular task that is taken up by him at this almost, I would say, around 10 days behind the scheduled date for his superannuation. It is really a bold step that he has taken up. And I knew right from the beginning, since he joined the department, that apart from his interest in teaching and research, another interest, if I am supposed to quote from my end, it is organizing and conducting the seminar, symposia, conferences, workshop. It was probably, I would say, another specialization of Professor Roy. So in that respect, he has very well, I was very much hopeful that he will be able to conduct this conference also in a memorable way, so that everybody in the department and every zoology family member, as it was put by Professor Roy, who might be outside the department, would definitely remember this particular conference. If I am to make one statement as to the overall the outcome of this particular conference, I saw very many papers, many times whenever conference was organized, the title of the conference and the papers presented do not go and match with one another. They are mismatched either way. But in this particular conference, it was very interesting. It was very nice to see that lot many papers, lot many talks, invited talks, were very much fitting with the theme of the conference. And because of the fact that the keynote speaker, Dr. Neelima Gupta, Vice Chancellor of the Bihar University, the talk, which was, of course, it was inaugurated by Professor Desai, my former colleague in the department, and subsequently, the first invited talk happened to be by Professor Home Chowdhury, head of the department of Calcutta University. And that particular talk, I was very much fascinated for one simple reason, that talk turned out to be fairly a good keynote talk for the matching with the title of the conference. Because that is, if those of you who might have concentrated on the talk might realize that he touched upon the match between the generation of the the nutrients and the footprint by the living fauna. If this match is not existing, if you keep on increasing, if one keeps on increasing the nutrient levels in any aquatic body, it may also increase parallelly with the increase in the living resources of that body up to certain level. And that much the catch will be available for human being to harvest. But when it crosses the limit, then there will be depletion of the living stock, and that will result into the excessive effort to be made by the human agency for getting the similar reward from that aquatic body. That is to say, that goes out of the sustainability level. And if you are maintaining the sustainability level, then we can enjoy any resource on this earth by entire human population, entire global population can take the advantage of this particular resource for generations. If this is not understood, and if there is a misuse and disuse of the resources by human population, then there will be sustainability level will come down. And that's the sustainability level coming down will be detrimental for the only planet to our knowledge holding the life and life of different type. I do not want to go into the details of other papers and all that, because report has been already given by Dr. Minal Desai. I can only say, to conclude my words here, that the overall conference was exceptionally handled in a much better and refined way. To my knowledge, there are around 15 invited talks and 20 oral presentation, 20 plus oral presentation, and 40 plus poster presentation. Everything went on very smoothly. And before I conclude my talk, I wish all the members of the Zoology Department, including, of course, the convener, Professor Roy, and Secretary, Organizing Secretary, Mrs. Desai, and all other members of the team, Dr. Evelyn DeCosta, who has been doing a good job of introducing all the guests almost all through for quite some time. With this, wishing all the best to the Zoology Department, I conclude my words. Thank you.
Thank you, sir. Thank you for your kind address. Over to Dr. Evelino Di Costa.
Thank you, ma'am. And thank you, ma'am. And thank you, Sir Shanbar, for that. Just a minute, sir. We're just facing some technical issues. I now request the participants of the seminar to come forward and share their experience about this virtual seminar. If anyone has any feedback that they would like to give to the seminar, to the organizing committee, we are welcome to hear it, whether it is positive, any kind of negative criticism as well is welcome.
Hello.
Yes,
Hello.
please go ahead.
I'm Dr. Hyacinth, Gujarat University, Ahmedabad. And I'd just like to congratulate the Zoology Department, the organizers, and all the staff and students for a very systematically planned, very well organized seminar, virtual webinar. Thoroughly enjoyed all the plenary lectures and the choice of the invited speakers, very, very informative, very interesting. And I also congratulate all the young presenters. Very well presented, very good quality of work. And I thank the university, Goa University, for this opportunity. And I look forward to their next venture. All the best. Thank you.
Anyone else for the feedback?
Thank you so much, Hailana.
Any of the invited speaker, if you all are on platform, you all can also give the feedback for these seminars.
Okay. Thank you, ma'am. If y'all can also type in your feedback in the chat box, and please do let us know what y'all felt about this virtual seminar. I'm sure I must have had a very good experience with all of the talks and received a lot of information from the invited speakers and the plenary speakers. We now move on to the valedictory address by our chief guest, that is Professor P.K. Sharma, Dean of the Faculty of Life Sciences and Environment, Goa University. Sir, you can take over, sir.
So please unmute yourself.
Hello, everybody. Can you hear me?
Yes, sir.
Okay. Thank you, Dr. Evelyn, for nice introduction at the beginning. And very good afternoon to everybody. Professor Roy, the convener of the seminar and head of the department. Professor A.B. Shanbhag, my former colleague. Nice to see you all there. Professor Roy, Professor Pai and Professor Shyama also there. My younger colleagues, Shanti Desai, organizing secretary, and Meenal Desai, Shamshad Sheikh, Savant, all of you in the family of zoology, and other delegates from various university and institute from the length and width of the India. It is a great pleasure indeed to be with you all. At the valedictory function of National Seminar on Life and Life Processes, an interdisciplinary approach for sustainable development. It's a very vast field and very apt for present time. And sustainable development is such a hot word these days, but lifestyle of people in developed world and upwardly moving people in developing nations is becoming more and more unsustainable. Present day lifestyle has become unsustainable, and due to it, we are facing regular environmental problems such as floods. You know, now even in Goa you get floods. Earlier you were used to hear in Bombay and other places. Now there are certain places in Goa this year, you know, where we have seen boats. So floods are there, droughts are there, forest fires you might be noticing if you go to the news channels. Potable water scarcity, more frequent cyclones, epidemics, and of course, the present going on pandemics. All these are basically problem with the non-sustainable lifestyle of we people. This I have mentioned from human perspective. What I just mentioned, all these problems which we are facing, it largely from the human perspective. And we are only one species. And now think about the survival of plants and animals and other organisms on this planet, which are also facing the consequences of our lifestyle. Basically, all these problems are because of the lifestyle. So what will be the consequences for all these organisms, and of course, in turn, that will also affect human progress. If this balance is disturbed beyond a threshold, survival of human will also become unsustainable. I remember a few years back, I was watching a international cricket match in South Africa, and that city where this match was happening, they were talking about that they have a potable water only for 1 week. And this is a very large city. I forget the name of the city. And I was shocked, and I was wondering, you know, maybe these are the kind of movement which people must have faced in earlier civilization, which we call, you know, Mesopotamia civilization or Harappa civilization. You know, these are the civilization which born near the rivers and may have extinguished because of some catastrophic nature which remove these water sources from there, or they might move from there, you know, like Gangetic civilization or so on and so forth. That might be just a migratory civilization now. Okay, so if we do not address this problem, which I am sure must have been addressed to some extent in this conference, it will really affect the well-being of human species as well. The exploitation of resources, or rather I should say over-exploitation of resources, the name of development, is resulting in damage to our environment. For example, let's take just one requirement, food. Now, food is not for sustaining ourselves. The food has become a pleasure also. And how it is causing our environmental degradation. The overfishing, okay, is a recurring problem, resulting in an aquatic food chain. Okay, overfishing is one thing, and then the fishing for, you know, what we call novel species, or, you know, exotic species. So whales are being killed, and all those, you know, big animal which has a very slow rate of reproduction. There overfishing is going on, and you know there are a lot of committees are there made by United Nations, but as in all democracy, these things work very slowly, and sometimes these committees are there, but no progress takes place. Okay, so this is exactly what is happening. Overfishing is causing a imbalance in the aquatic food chain, and related consequences of disappearing of animal biodiversity and health of water bodies, etc. Second is growing food for increasing population. Now, if the productivity is not sustained to meet the requirement of the people, then there'll be scarcity, there will be, famine and things like that, maybe riots, which happens. So to overcome that, we have to produce more, and to produce more means what? We just have to put fertilizers, those new species and genetically modified things are, still in the pipelines, especially in India. The rest of the world have moved ahead. We are still, discussing the regulatory aspect of that. So growing food for increasing population also resulting in stress on water tables, use of excessive fertilizer to soil degradation, with its apparent consequences to the rapidly increasing population. So we will not be able to meet the requirement for food in near future. Of course, technology is always there, which I will come upon. Zoologists might have already heard the terms, artificial meat, lab meat, and that meat is already available in the market in the developed country. So now we have to start growing meat in the laboratory also. That will be a great field to work by my younger colleagues in the zoology department. Need for exotic food, that is another thing, results in additional cost on account of its transportation. That is burning fuel. Now you see, if you have a money, you would like to have a particular food, which is, suppose you are in California, but the food is grown in, say, India or Bangladesh or Sri Lanka or Japan. And this food will be flown to the people who demand that food. So this add additional cost because of exotic food, because of non-sustainability, because we are not satisfied with what is local. Okay? So that is another addition to this what we call non-sustainable lifestyle, and which is affecting our environment. So exotic food results in addition of cost on account of its transportation to a long distance, say some point in Eastern Hemisphere to Western Hemisphere and vice versa. Our ancestors had such a sustainable lifestyle, a beautiful sustainable lifestyle our ancestors had, that their needs were met from local produce, causing minimal resource exploitation. And that is where we all need to look for involvement of local community in all our needs. We need to focus local for long-term sustainable goal and need to stabilize population. Of course, that is another hot topic in India. Population also become issue which should not, which relate to everything else. This is a subject which is truly interdisciplinary. This what you have discussed in this virtual conference, life and life processes, sustainable development. So this is really an interdisciplinary subject. And of course, I was going through the report and I saw that you have a plenty of presentation, 20 oral presentation and 12 resource person, and most of them were in the field of zoology, animal sciences. Okay. However, this is a field which need to have a conference, which should be addressed by multidisciplinary people like technology, some people from society itself, policymakers, educationists, environmentalists. In fact, everyone has to work towards sustainability for well-being of every living being, be it animal, microbes, plant, human, or our ecosystem. I learned from the comments of participants, and of course, Professor Shanbag also mentioned some of the highlights of some of the lectures, that during this seminar, you had a great learning of these processes required to maintain life and life processes. And without taking any more time, I take this opportunity to congratulate the Department of Zoology for organizing this virtual seminar in these very difficult time and to continue such academic activities, which is very, very important. I'm really, like to thank you and congratulate you for this activity. I understand Professor Roy will be superannuating at the end of this month. He may like, as as Professor Shanbag is mentioning, that he has been part of various organization of various seminars in the department, and subsequently bringing out the proceeding of those meetings. I hope he may like to take up coming out with proceeding of this seminar as well, which will benefit the youngsters and everybody in the field. I take this opportunity to wish you all a good health and great academic progress in future. Thank you very much for inviting me for this function. Thank you.
Thank you so much, sir, for your inspiring words. Thank you, sir, for your inspiring words and motivation for the department. I now request Dr. Shanti Desai to propose the final vote of thanks.
Good evening, everyone. This is Dr. Shanti Desai, Organizing Secretary of this National Seminars on Life and Life Processes, Interdisciplinary Approaches for Sustainable Development. I take this opportunity to propose and record vote of thanks for this valedictory program. I, on behalf of organizing committee of this national event, would like to put on record our sincere vote of thanks to our Honorable Vice Chancellor, Professor Varun Sani of Goa University, who had graced our inaugural program. We also thank him for all timely administrative support and encouragement to organize this national event. We put on record our sincere thanks to our Honorable Vice Chancellor, Professor Neelima Gupta, Vice Chancellor of Tilka Manji, Bhagalpur University, Bihar, who had graced our national seminar through her address this afternoon. She could not make it for first day for inaugural program as a keynote address, so we had it as a concluding address for the national seminars. I would like to put on record our sincere thanks to Professor Radhika Nayak, officiating registrar of Goa University, who have facilitated us to organize this event. We also place on record our thanks to Professor P.K. Sharma, Dean, Faculty of Life Science and Environment, Goa University, for gracing this function as a chief guest and for his valuable address. On behalf of organizing committee, our hearty thanks to Dr. A.B. Shanbag, the ex-HOD and Professor of Department of Zoology, Goa University, who honored us by gracing this valedictory function through his presence and kind address, and for accepting our invite at a very short notice to be a guest of honor for today's function. We also thank Professor Sanjeev Gadi sir, Professor Sandeep Gar sir, and Professor Krishnan sir, respective heads of the biotechnology, microbiology, and botany department of Goa University for judging the poster presentation. Our sincere thanks to all invited guest speakers for their plenary talks, invited talks, and for chairing the session. And I thank all the participants for such overwhelming response. We thank them for all of their great share what they have for making this virtual national seminar a great success. Our heartfelt thanks to all our ex-HODs and professors, uh, Professor Paranjape sir, Professor P.V. Desai sir, Professor A.B. Shanbag sir, Professor S.K. Sharma sir, and Professor I.K. Pai sir for accepting our invite for the felicitation program and honoring us by their presence. Some of them were unable to make it today due to connectivity issues at their end. Shri MP Tonsekar sir, the coordinator, we also thank him. He could not make it today for this function, but our sincere thanks for him also, because he had initiated our department as a coordinator on deputation. We also place on record our thanks to Professor Janardhan sir, a director, Directorate of Digital Learning and Initiative, and his team members, Sri Sachin Sambaji, for live streaming of this event for 3 days on ZooSam YouTube channel, since we were crossing the participants above 100. We also put on record our thanks to Project Office, the computer section, and other supporting administrative staff of the Goa University for the needful support to give uninterrupted internet connectivity and power supply at Council Hall for our show to go on in virtual platform. Our sincere thanks to our MSc student Divya, Prajwal, Shravya, Karthik, and Shwain for being here at Council Hall as a volunteers for all 3 days. A strong team with a good leader is must for sure to move on. And with that, I thank our HOD of the department, Professor R. Roy, sir, for his due blueprint of this event and for which his team has executed in well-planned manner. And for that, I thank Dr. Nitin Sawant, Dr. Meenal Shirodkar Desai, Dr. Avelino Di Costa, Dr. Shamshad Sheikh, and Ms. Gandita Kundaikar, assistant professors at Department of Zoology, Goa University, for coordinating throughout for the smooth conduct of this national event. My special thanks to Ms. Gandita Kundaikar and Dr. Avelino Di Costa for compiling the abstract book and certificates, the hard copies of which will be posted to all our participants and invited guests. And now I thank our non-teaching staff, supporting staff of the Department of Zoology, Goa University. If I have missed anyone, then I beg your pardon. Thank you one and all. And we look forward for fruitful interaction leading to research and academic collaboration in near future for expansion of the knowledge to enable youth-inspired research for years to come. I now request all to put on their video for a screenshot for our record. I kindly request all the guests who are present on this virtual platform to kindly put on their videos. I'll be taking 3 shots. I'll just go for a count of 1, 2, 3, and then we'll go 3 times and we record it for our national seminars. We'll take the first shot now. 1, 2, and 3. I'm changing the participants. Maximum the better. We'll wait for 1 or 2 seconds. Yeah, the second one. 1, 2, and 3. And the last one, with the wide smiles on everyone's face, we go there as 1, 2, and 3. Yeah, that's it. I call upon Professor Roy, convener of this event, to declare the closure of these national seminars.
I, Dr. R. Roy, as a convener of this national seminar, I declare the seminar is over. Thank you.
I now request all to kindly raise for the national anthem.
Himachal Yamuna Ganga Uchala Jaladi Taranga Tava Shiva Name Jage Tava Shiva Ashish Mage Gaye Tava Jaya Gatha Janagana Mangala Dayaka Jaya He Bharata Bhagya Vidanta Jai Jai Jaya He, Jaya 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 He.
Thank you, everyone. Have a good evening ahead.